Hello, so this is going to kick off our lecture for chapter 3, and it's all about the organic molecules of life and the macromolecules we make using them. So we're going to talk about carbon molecules, a little bit about how carbon can be configured. Throughout the series of videos, we'll also discuss some of the bonds, and then we will talk about carbohydrates and lipids. Following that, we will also talk about proteins, and then also nucleic acids. So they're not listed here, but they will be something that we definitely cover throughout the video series. All right, so the first thing is this whole term organic. I know you've heard of the saying of something being organic or not. If something is organic, that means it contains carbon. So a lot of living organisms, of course, are organic because we contain carbon. That was one of the top 4 elements that all living things are made up of. An inorganic molecule does not contain carbon, so it's usually something we make or synthesize, and because of that, if it does not contain carbon, we call that inorganic. So like I said, there are 4 elements that make up living things we talked about in chapter 1, and they are CHON. So carbon, hydrogen, oxygen, and nitrogen. Those are all going to make up those living things. Okay, so here we have the carbon atom. Remember that number 6 is going to be our atomic number. That's going to tell us our number of protons and our number of electrons. And then we have an atomic symbol, of course. And then finally at the very, very bottom, we're going to have our atomic mass, which is going to be the number of protons plus neutrons. So seeing this information and understanding the drawings we've been doing, you should be able to tell me how many electrons do we have in our valence shell, and then furthermore, how many bonds can this carbon molecule form? So think about that for a second. What you should have came up with is something that looks like this, in which we have 4 electrons in our valence shell. And then because we're working with the octet rule, we have 8 minus 4. That means we have 4 bonds that we can actually create with carbon. So we have another bond for an atom that can go here, another atom can go here, another atom here, and another atom here. Of course, I am drawing this considering our orbitals have space for 2 electrons, so I filled in the extra black electron here, and you can see the 4 additional bonds we can form. So the great thing about carbon is because it has 4 bonds, carbon has the ability to create single bonds in which you share 2 electrons, a double bond in which you share 4 electrons, and a triple bond in which you share 6 electrons. You can see the structural formula. You remember letters and lines that I talked about? You can see the structural formula here, in which it includes the letters and the lines. Again, this is just another image showing how carbon can form all of these bonds. Also, there's different configurations. I won't ask too many questions here, but you may see it in chemistry courses in which carbon can form these ring-like structures. It can form highly branched structures, and also things that are going to be more linear, more like a line. So one special thing about carbon is it's kind of cliquey, right? It likes to kind of hang out with its own friends, and carbon tends to bind with other molecules that are part of CHON. In particular, carbon really likes to bond with other carbons, so we tend to have very long carbon-carbon chains in real-life scenarios in which carbon will form long chains of binding with other carbons. A lot of times these carbons also have extra bonds with hydrogens, so sometimes we draw the hydrogen, sometimes we don't. But as you see, every single C that I have on the screen has at least 1, 2, 3, 4 single bonds, right? So 1, 2, 3, 4. 1, 2, 3, 4. It all has those bonds around the carbon. Carbon also has the ability to form both polar bonds and also non-polar bonds. So remember, polar is going to have a little bit of a charge because one of the atoms has a higher electronegativity. And then nonpolar is usually going to be no charge, right? This is something like an oil that does not have any type of charge. Hydrocarbons are going to be predominantly carbon-carbon bonds or carbon-hydrogen bonds. Hydrocarbons is just what I drew earlier, in which you have long chains of the carbons, and then on the sides you have those hydrogens everywhere. Asking yourself if this is polar or non-polar, it's actually going to be non-polar. So these molecules are hydrophobic, meaning they have a fear of water. That's a great telltale sign to let you know that it is a non-polar interaction there, because hydrophobic molecules tend to be non-polar. Some of these you may have heard before: methane, ethane, propane, anybody's on a grill, or butane. So these are all examples of hydrocarbons. Again, carbon can form interactions with oxygen or nitrogen, and when it does that, these tend to be polar bonds, meaning it's going to be hydrophilic, but some portions of it can also be hydrophobic. So it really just depends on where the interaction is. So for example, carbon binding with oxygen, you see oxygen has a bigger arrow, a bigger pull on electrons because it has 8 positive protons versus carbon 6 positive protons. That means it's going to have a higher electronegativity here. Another thing I wanted to mention was a functional group. So a functional group is going to be groups of structures that have very similar properties. So it's going to be almost like an addition, right? So we have our carbon with our hydrogens, with our hydrogens, with our hydrogens, and then you have this little functional group. And the functional groups have certain characteristics. So great examples are things like hydroxyl groups. You may remember this from pH stuff. Phosphate groups. Maybe you've heard of ketones for a keto diet. That is a functional group. So I don't want you to understand all of the functional groups by name. You don't have to do that. I just want you to understand the definition of a functional group, in which it shows that if it has the similar chemical structure, it will have some type of similar chemical property. So this is kind of just a quick overview about carbon, because we're going to be talking about some of the cool things carbon can do throughout the duration of the video. So let's talk about synthesis and breakdown. Very, very important. I'm going to introduce some terms, and we're going to talk about how we can build something up and how we can break something down. So with a monomer, introducing some vocabulary here. Mono actually means one. So I don't know if you've seen a definition or a word that has mono in it, but mono means one, okay? They're going to be representative of the smaller molecules. A monomer is going to be one unit of something, okay? If you take multiple monomers together, string them together, you have a polymer. Poly means many. So a polymer is a chain of monomers. So I'm going to do the M to let you know each individual unit is a monomer, and collectively they are known as a polymer, right? This is a very important concept because we're going to be talking about going back and forth between monomers here, which are the individual little, P's in the picture, how you string them all together and you make a larger unit called a polymer. So how do we build that larger unit called a polymer? We build that by a process known as dehydration synthesis. Dehydration synthesis. It's very important that you understand dehydration synthesis is for building. It's for making. Synthesis is your key word here. To synthesize something means you made it. Think of like synthetic hair. Synthetic hair is fake hair. It's hair that we made, right? A synthesis is something you make. Dehydration, you know what that means. That means without water. Without water, or in this case, removing water. That's going to be a hint for you moving forward. So we know dehydration means removing water, taking out water, without water. Synthesis means to make or to build. So this is how we build our polymers. How do we do that? Our monomers are going to be joined together by the components of water, which is H2O. So what happens is we are going to remove the H2O from our monomers. We're going to take an OH group from one monomer and an H from another one, which will form H2O, and this helps us to make a polymer. So again, monomer 1 has an OH hanging from it. Monomer 2 also has an OH from it. We find the water pieces, which are OH here. We remove them, and you're left with one remaining O, which will actually link together your bond. Water is a byproduct of the reaction. So a byproduct is something that we don't want, but we get as a result of completing the reaction. And this H2O is going to be released. It's going to be exiting. Do you see this arrow? I'm going to draw all of this on a separate slide, but I want to walk through the concept now. The monomers are linked together, and this process can happen over and over and over again. So let me draw this out for you so you have another visual. So dehydration synthesis, okay? What happens here is we have monomer 1. I'm just going to say M1, and I'm going to give you another color to have monomer 2. We're going to do an OH here because all the monomers have OHs hanging off of them. So it's an OH on that side. And the second monomer also has an OH. We want to add these together. So at this point, we have one bead. We want to add it to the other bead, right? We're trying to go from a monomer to eventually forming a polymer. That's the whole concept. What happens here? As we move forward in the reaction, we are going to remove the water. Do you see how if we take an OH from one group and an H from the other group, we actually end up with H2O, 2 H's, 1 O? That's what happens. That H2O we create is going to leave, H2O. When it's left, all we have is this remaining O. So what's next? We still have M1. We have M2. And now we have this little O that we can connect the two of them together. Let me redraw the O in a darker color. Boom. Now we formed our polymer, or we're starting to form our polymer. This process can happen over and over and over again. So let me just walk you through what's here. Dehydration synthesis is where we remove water to build something. We have two monomers. Both of them have OH groups hanging off. There's one, there's two. By removing water, water leaves with an arrow. The water comes from here. We can connect monomer 1 and monomer 2. We're going from single monomers, we're building a polymer. If we remove the H2O, we just have one O that's left. That O can now be connected to build up monomer 2. Okay? If that is confusing, then definitely rewatch this portion of the lecture. Dehydration synthesis here, this is just an image from the book, kind of re-showing that. So they kind of got rid of our O here, but again, OH, OH, you remove the H2O, now you can link these together. So that's how you build up a polymer. Let's talk about breaking down the polymer. So breaking down the polymer occurs through a process called hydrolysis. Hydro means H2O. Lysis means break. So the word literally means H2O breaking. So H2O is going to break something up. And if you learn dehydration synthesis, this is just the reverse reaction. So now we're going from having a polymer and trying to break it down into individual monomers. So water breaking. The addition of water, adding water, is going to break up or lyse the bond. So water is required for the reaction. If you have no water in this reaction, you have no reaction. Water is required. This helps to split apart monomers. This can happen over and over again. So again, if we're looking at this image, this is going to be our polymer. They're linked by that O. By adding in the H2O, you're now adding an OH to one side, and that remaining H goes with the original O that was there. So let me draw it out just to give you another example of seeing this live in action. Hydrolysis, water breaking, going from polymer to monomer. What do we have? We have monomer 1, we have an O linkage, and then we have monomer 2. In this reaction, here you're going to see H2O enter. So there's going to be an arrow going in. We're not going out like dehydration synthesis. We're going in and we're adding H2O. What's next? Well, we still have monomer 1. And we still have monomer 2. Now it doesn't matter where we give this O to, because the O can go to either monomer 1 or monomer 2. It literally doesn't matter. You can change it. All you want to make sure you do is add one of the H's from H2O to that O that was there, and then the other side you can add the remaining OH. So now this is showing us going from a polymer over to two separate monomers. Hydrolysis. Again, this is an image from the book. Not a big fan of it, but still wanted to show it. If you are confused with hydrolysis or hydrolysis and dehydration synthesis, please review it. You may have noticed, but hydrolysis, going from the combined substance, adding water, you get two separate monomers. It's just the opposite of dehydration synthesis, where you're going from the two separate monomers, you're removing water, and then you get a combined one. So it's literally the same thing. This is dehydration synthesis. If I wanted to quickly draw hydrolysis, I'll draw it on the bottom. Please, don't get confused. Hydrolysis is breaking it down, right? This is where we break it down. We're going to go from M1, circle, M2, let me redraw that for you, M1, circle, M2. In this case, we're adding water, H2O, adding it to break it up. That's going to give us M1 OH plus M2 OH. It is the opposite of one another. So these are going to be the opposites of one another. Please make sure you review dehydration synthesis and hydrolysis so you're very familiar with these concepts. Okay, so kind of thinking about you are what you eat, trying to live healthy lives. We're going to be talking about different food groups and how we break down those foods and what macromolecules they make. So keep in mind, we all know this. When you eat your food, you're going to digest it into smaller pieces. Your body then takes those small pieces and builds up macromolecules that you can use to be a functional human being. So think about it. Maybe you know, maybe you don't. What are the 4 classes of macromolecules or biological molecules? Hopefully you identified. We have carbs up top. We know carbs. We love carbs. we have proteins, we have lipids, which are like fat, and we don't have a picture of it here, but we also have nucleic acids. So those are going to be your 4 macromolecules. We're going to pause this lecture and then we will pick up with carbohydrates.